Imagine a world where we can regrow limbs, where lost organs can be replaced with lab-grown ones. It sounds like science fiction, but stem cells make this a possible future. Stem cells have two properties that make them so special and unique. First, they can divide indefinitely. Whereas other cells have lifespans of weeks, months, or years, stem cells, when kept in the correct conditions, can live forever. And secondly, stem cells have the ability to form any other cell in the body, a property called pluripotency. Think of it as a ball rolling down the hill. At the top of the hill, it has the potential to go down many different paths and end up in many different places, perhaps skin, heart, or brain. The further it rolls, the more specialized it gets, and once it starts, it keeps going. If it starts down the pathway to heart, it stays heart. So technically, we can take some stem cells, grow them until we have sufficient numbers, then change the conditions they're in and turn them into any cell type we want, and use those cells to engineer tissues for use in medicine. Amazing, sign me up. There is a catch though. To isolate human stem cells requires a human embryo. This means obtaining material is difficult, and even then, there's a massive ethical dilemma of using a potential human life for research. So much so that in the US in 2001, George W. Bush stopped all federal funding on human embryonic stem cell lines. It seemed then in the early 2000s like the promise of this field was lost. Enter the Yamanaka lab with enterprising postdoc Kazutoshi Takahashi and the head of the group, Shinya Yamanaka. They would revolutionize stem cell research. Their thinking was simple. We know the unique factors present in stem cells that make them stem cells. So what happens if we take another cell, say a skin cell, and induce the expression of those unique factors in it? Could we turn that skin cell into a stem cell? They identified 4 crucial molecules, now known as the Yamanaka factors, that, when introduced into an adult skin cell, turn them into cells with all the properties of embryonic stem cells. They called them induced pluripotent stem cells and published their work in 2006. This started a massive movement in stem cell biology. Instead of needing embryos, we can create stem cells from skin. That ball at the bottom of the hill had suddenly developed wings, and it can fly back to the top. An unheard of idea, the need for embryos completely circumvented. But the science is still in its infancy, and the efficacy and safety of these cells still needs to be rigorously tested to ensure they're safe for you to use in humans and can do everything that stem cells can. Even so, the promise of the stem cell field, once thought to be lost, has been given a new lift by the Yamanaka method of iPS cells. Thank you.